Hemoglobin, the major protein found in red blood cells, is responsible for transporting oxygen from the lungs to tissues throughout the body. Hemoglobin is a tetramer made up of 4 polypeptides, 2 alpha subunits and 2 beta subunits. This tetrameric structure is essentially a dimer of alpha-beta heterodimers. At the core of each subunit is a heme complex made up of a porphyrin ring with a central ferrous iron atom. The iron has 6 coordination bonds, 4 with nitrogens in the plane of the porphyrin ring, and 2 with histidine residues above and below the ring called the proximal and distal histidines. In the absence of oxygen, the heme group is puckered because of the large radius of the ferrous iron atom. Oxygen binding to the heme group reduces the radius of the iron, forming a planar heme. Because of the bond between the iron and proximal histidine, movement of the iron moves the histidine, which in turn moves a helical segment called the F helix of the hemoglobin polypeptide. The F helices are located at the interface between subunits, and therefore the movement of the F helix of one subunit can impact the structure of the entire protein, shifting the hemoglobin molecule from one conformation, called the T state, to another conformation called the R state. Oxygen acts as a positive allosteric effector for hemoglobin by shifting hemoglobin from the T state to the R state. Several other molecules act as negative allosteric effectors. Their binding shifts the conformational equilibrium towards the T state. The small compound 2,3-bisphosphoglycerate, or 2,3-BPG, traps hemoglobin in the T state by binding between 2 beta subunits in the center of the hemoglobin tetramer. In this way, 2,3-BPG acts as a classic heterotropic negative allosteric regulator. Binding of 2,3-BPG to a secondary site prevents binding of oxygen to the primary site. 2,3-BPG plays a role in facilitating oxygen transport from maternal cells to fetal cells during pregnancy. Although 2,3-BPG is present in both adult and embryonic red blood cells, fetal hemoglobin consists of a tetramer complex in which the normal adult beta subunit is replaced by a special fetal hemoglobin subunit called gamma. The fetal form of hemoglobin has a lower affinity for 2,3-BPG. As a result, fetal blood cells have more hemoglobin in the high-affinity R state conformation compared to maternal red blood cells, and therefore fetal hemoglobin can acquire oxygen from maternal hemoglobin. The multiple modes of hemoglobin regulation ensure that oxygen is efficiently delivered to tissues. Hemoglobin also serves as a classic example of how protein structure mediates protein function.